When I write a sequence like this, does it look like a secret code to you? Don't worry, by the end of the video, you will read it like a DNA sequence. Hello everyone, welcome back to my channel Genius Minds. In today's video, we are going to learn the basics related to the DNA strand, and it is going to be very important for the CBSE 12 board students. So let's get started. When we talk about DNA is made up of small nucleotides. It is made up of nucleotides. Now, what are these nucleotides? Nucleotide comprises three important things. The first one is the sugar molecule, the phosphate, and the nitrogenous base. Very important in biotechnology, the nitrogenous base. The sugar which is present in DNA is known as deoxyribose. And when we talk about the nitrogenous bases, there are four important nitrogenous bases which are represented as ATGC, where ATGC, where A stands for adenine, T stands for thymine, G stands for guanine, and C stands for cytosine. So your entire DNA strand is made up of these nitrogenous bases, along with the bases sugar is there and the phosphate molecules are present. Now when we look into the base pairing, which is very important topic, so there are some rules what these nitrogenous bases follows. So as we all know, DNA is a double-stranded molecule. It is a double-stranded molecule where all these bases are present, whatever we have studied, A, T, G, C, randomly in any sequence. Now, how are they going to form the base pairing with one another? So it is said that the adenine molecule always pair with thymine. G always pair with C. That means adenine with thymine, G with C. So if you will see the base pairing, and in the another strand, according to the rule, the pairing will be T, G, A, C, T, A, G, C. This is known as complementarity rule. This pairing is known as complementarity rule. So this is how the bases are paired. Now when we look into the another important part, that is the importance of the enzymes called as restriction enzyme. So it is very important to know the basics of DNA before moving into the restriction enzyme. So now these restriction enzymes, they play a very important role in the rDNA technology, recombinant DNA technology, where they are responsible for making specific cuts in this nucleotide chain. So whatever the DNA strands we have seen now, so they are very specific where they are going to cut the strand. So this is all about the restriction enzyme. So what are these restriction enzyme and why do you want to cut our DNA? That all we are going to cover in our next video. So stay tuned. If you are new to my channel, please subscribe it, share it. Thank you very much.